So our format for today is we're going to be leaving all Q&A until the end of all of the panelists. So Chris, come on up, please.
Thank you very much. That was a wonderful introduction. I'm going to talk a little more on the hazard side of things. But as I go through this, keep in mind exposure is a very, very important component. So thanks, Charlene. This is our institute. Brian Civic indicated that there were 27 institutes with the National Institutes of Health. There's one that's not here on campus, and that's the Environmental Health Sciences, the National Institute of EHS, and this is our institute down in Research Triangle Park. This is our mission, to discover how the environment affects people in order to promote healthier lives. So hopefully very relevant to the discussion today, and I look forward to answering some questions. This is our vision. We follow our mission through a multifaceted approach involving fundamental research on exposure, which I'm not going to talk much about today, translational science. We're very interested in applying what we do to your problems and environmental health problems in general. We're global in reach. We have a large global environmental health program. It's actually operated out of the office here on the campus, our small office where in fact I work. And we participate in training. Again, another topic that Brian touched on this morning. We're very involved with training environmental health professionals nationally and internationally. I want to talk a little bit today about something that's relevant to you and try and give you some translational information to help you make some decisions. I'm going to talk about a couple of projects that we have, and we are very involved with right now. We do a number of things, hundreds of things, but I want to just focus on a couple of things that I think are perhaps interesting and applied for you folks. We're going to do this very quickly. You're going to run through some endocrinology, some physiology, some toxicology, some biochemistry, and a little bit of chemistry here in the next two or three minutes. So fasten your seatbelts. There are about 85,000 chemicals on the market today. Not all of those are toxic, thank goodness. Only a handful of those have been adequately tested toxicologically. Literally, only a handful of those chemicals have been tested. I want to talk about a group of chemicals. Not all of those chemicals are endocrine disruptors, but we're aware that some of those are, and we're looking feverishly for those that are, and I'll explain how we're doing that in a minute. You may look at this list up here. You don't need to memorize this. We're not going to test you on these, but you may recognize some of these chemicals as being under your sink, in the building materials that you use, etc. Many of them are insecticides that are used commonly in buildings, probably here at HHS, NIH, industrial chemicals, etc. Plasticizers, bisphenol A is very controversial right now. These are some of the endocrine disrupting chemicals that we're aware of, chemicals that have the potential to disrupt the endocrine system, which of course guides development throughout life. We know that anybody that's raised a child or two or three knows that development occurs across a wide variety of the lifespan. It begins actually before conception, and through conception and prenatal development, postnatal development. I've got a 24-year-old son at home. He's fortunately his frontal lobe is just now becoming well developed. I've waited 24 years for that. So the point is that development occurs throughout life. It's common sense. But one of the most important parts of development is when the cells are dividing very, very rapidly. And children are growing, they're learning, they're absorbing all kinds of things in their brain. They're also absorbing things from their environment. And we're looking very closely at that aspect of development. We now know that there are windows of susceptibility, biochemical susceptibility, that open up, some for days, some for a few months, some for 24 years. But there are windows of susceptibility as humans develop. And that's particularly important very early in the developmental phases, in utero and postnatal. We now know that those windows of susceptibility can open, an assault can occur, a chemical assault or an endocrinological assault can occur during that window of susceptibility. The window closes, the disease doesn't appear for until later in life. Sometimes a year later, sometimes 2 years later, sometimes 40 or 50 years later. Think of mesothelioma. Anyone that's watched late night TV knows that mesothelioma is a disease that doesn't, your exposure can occur early in life. It doesn't appear until much later in life. So that's an area of interest to us. Here are some examples of diseases that we know, and you probably know someone, if you don't have a family member that suffered one of these diseases, that we believe is a result, at least partially, of an exposure that occurred during one of those windows of susceptibility: breast cancer, fibroids, obesity. And I'm going to talk a little bit about obesity. This is an experiment by way of proof of principle. We know that these endocrine disruptors can cause even short exposures. In this case, this is an animal study using rats, where in the red line here, the animals were exposed for 5 days to a known endocrine disruptor. In this case, it's DES, or diethylstilbestrol. And those animals, so they were exposed for 5 days. The exposure stopped. Both the control animals and the test animals ate the same diet, exercised exactly the same amount throughout the course of their life, and one became obese. One set of animals, one group of animals became obese. The other did not. So we know through proof of principle that endocrine disruptors can cause this type of problem. And there are a wide variety of similar lifelong problems that you might be aware of that I won't really go into right now. So our institute is involved with identifying some of these toxicological problems, characterizing those, sharing that data. Again, harking back to what Brian Sivak said, we're very interested in data sharing. You have access to the data that I'll describe here in a minute. We don't want to just identify these problems. We want to help you solve these problems. We've developed a program called Toxicology for the 21st Century. This is an evolving program of research. It's been underway for about 10 years, but it is really changing the way we think and the way we act. Instead of testing animals retrospectively for disease, what we want to be able to do, ideally, is predict disease based upon chemical behavior at the metabolic level. So just north of here, we have a battery of robots. These are robots that were modified from the manufacturing of automobiles. They're 3-armed robots up in Rockville, Maryland. And 24 hours a day, 7 days a week, they're running biochemical assays on on cells that we think are at least partially predictive of metabolic processes. So if you look at what we call a heat map on the right side of the panel there, across the top, it's not important necessarily right now what those chemicals are, but across the top there may be 50, 100, 5,000 chemicals. We can test up to 600,000 chemicals a day with these robots in a variety of cellular and biochemical assays, which are plotted down the left-hand side of that heat map. So, if you have a chemical, for example, you see the red streaks along the right. Let's say those are all biochemical or cellular assays that are indicators of estrogen activation. So if you have a chemical and you are concerned about its toxicity, and you have an alternative chemical, you may choose the one that doesn't light up all of the metabolic pathways associated with endocrine disruption. So, and you can test, in this case, I use the example of estrogen toxicity, but you could do that with almost any type of panel. Now, this is a developing process. It's not ready for prime time. It is being used for deciding which chemicals we really want to use more expensive toxicological testing on, but it's a program that we hope ultimately will help you decide which chemicals you may want to use in building materials versus others. This is just a panel to show that we're able to use this same method to test a wide variety of concentrations. We are concerned about actual exposure, relevant exposure, translational exposure. And we can run these batteries over and over, so we want to make sure that they're repeatable, certainly, as well as run across a wide variety of materials. So with that, the question that I posed, that I turned in was, how do we use red lists, cell-based screening tools like the one I just showed you, and alternative animal models, which I didn't speak about today, but we do have alternative animal models such as the zebrafish, which is a wonderful model for developmental toxicology. How do we use these models to address individual compounds in a complex real world of mixtures? So thank you.